All right, welcome to this episode of Lectures with Liddington. This is our second episode about biochemistry. Before we dive into all the different specifics of the different macromolecules and what they do, their functions, we really have to talk about some basics about macromolecules, what they are, and how we make them and how we break them down. So that's what we're going to talk about in this episode today. So macromolecule. What's a macromolecule? Macro meaning large. So last episode we talked about carbon and how carbon can make lots of different types of molecules. It's so variable because it has those 4 electrons that it can share. So we can make lots of different kinds of macromolecules, meaning large. In living things, we can break down these macromolecules into 4 main categories. They are carbohydrates, lipids, or fats is the other term for lipids, proteins, and nucleic acids. Because these are biological macromolecules, sometimes we refer to these as biomolecules. So when we talk about making these big molecules, there are some other terms that we use for those pieces. Really big molecules we make by taking smaller pieces and putting them together. So these smaller pieces, these are called monomers. Think mono meaning one. So a monomer is one piece that you can put together to make a larger piece. That larger piece is called a polymer. Poly meaning many. Think other terms like polynomials or polygon, right? If you go into math, poly meaning many. And a polymer is basically just a large molecule made of lots of little smaller pieces, those monomers that we've put together. Okay, well, how do we do that? How do we make these macromolecules? And we do that through a process called dehydration synthesis. To synthesize is to put together. So that's what this means. Synthesis is to put together. And dehydration is literally taking away water. So that is literally what's happening when we are making a macromolecule. We put together a large molecule out of little pieces, out of those monomers, by removing water. You'll take a hydrogen off of one side, and then you'll take an OH, that hydroxyl group, off the other side. Those 2 pieces will come together. Notice we've got one H here, one H here, and one O. H2O, right? So I've taken those 2 together. They come together as water, and they leave as water. It's the love story of water. Dehydration synthesis is the love story of water. That hydroxyl and that hydrogen come together, and they run away as water. So I now have my 2 monomers together as one. Let's see, what does that look like? So in this picture, in this diagram, we're showing we have one monomer over here, right? This little circle is representing whatever subunit this is. Just a very general picture. They could be carbohydrate units, they could be proteins, whatever it is. It's some subunit, it's some monomer. And over here is a chain, a little polymer that I've created previously, and each one of those is a subunit. But I want to take these and put these together. So notice here I have my OH, my hydroxyl. Here I have my hydrogen. 1, 2 H's, H2, 1 O, H2O. These 2 will come together and then run away as water. Now that you've broken those off, you have these two spots here. I've got two molecules with an open bond, with an ability to bond. So they will bond with each other. There's that bond created when those two pieces come together. Let's take a look, not with that general circles picture, but with more specific. Each one of these is a carbohydrate monomer. This is a glucose. Notice over here, I've got this hydrogen chilling out here. I've got this hydroxyl chilling out here. They see each other and they run away together, woo, as H2O. Then these other two, there's an empty space. So they will now bond together, and I will end up with two glucoses bound together. I now have two monomers together to make a very short polymer. You could do the same here with my protein. Notice I've got an OH over here, I've got an H over here. They will run away together, and instead I will end up with a bond here between that carbon and that nitrogen. The opposite, when I want to break down a molecule, think about it. You got this huge molecule. How are you going to get it into a cell? It needs to be smaller to get in. So I've got to break it down. And I do that through a process called hydrolysis. If we break down the word hydrolysis, hydro is water, and lysis means to cut. So I am literally cutting a molecule with water. I'm breaking down a large molecule by cutting it with water. I take a water molecule and I smash it into that bond. Hydrogen goes to one side, OH goes to the other side, and they are now broken apart. I have completed those bonds with those pieces of water, and now I've got smaller subunits. What does that look like? Let's take a look. Let's move this here so you can see it. So up here I've got my polymer. These are my subunits. So I've got 1, 2, 3, 4, 5 subunits in my polymer, 5 monomers. So what happens is I have water, and water will come in and it will bombard that bond. It will cut it, right? Lysis is to cut. It will cut that piece right there, and then I'll have one of my hydrogens will bond on the one side of the one polymer, and then my other hydrogen and the oxygen will bond over here with that monomer that I just lysed, I just cut off. So let's test your knowledge and see if you can try it on your own.